students. In the previous class, we have discussed the advantages of the GM plants, right? So, what are the advantages of producing the genetically modified organisms we have seen? Now, let us see some of the applications of the biotechnology in different fields. So, the first area where the application of biotechnology is very much high rate, that is in the areas of agriculture. So application of biotechnology in agriculture, we are going to see today how the biotechnology is helping in the agricultural areas to develop the genetically modified crops having different properties and different qualities. We have seen the advantages of genetically modified crops, that is making the crops tolerant to abiotic stresses, reduced reliance on chemical pesticides, right? Enriched nutritional value it is having, and helping the crops to utilize the maximum nutrients supplied to it. These are the some of the applications of the biotechnology in agriculture, in specific, that is advantages of the GM crops. Now let us see how the genetically modified organisms or the genetic engineering helping in the agriculture. So one of the best application of biotechnology in agriculture is production of insect resistant plants, that is production of insect resistant plants. So one of the advantage of the GM crops what we have seen is reduced reliance on chemical pesticides. Yes or no? So after the green revolution, what were the drawbacks of the green revolution? One of the drawback is the chemicals that was being used in the agriculture, that is the agrochemical based agriculture is going to produce lot of chemicals or the toxins in the environment, which is producing pollution in the environment. So therefore we want reduce usage of these chemical pesticides. So to overcome that issue, the biotechnology has produced insect resistant plants. Means now we are using the pesticides and insecticides to kill these insects and pests. If the plant itself has the inbuilt resistance towards these insects and pests, then external use of these chemical pesticides can be reduced. So there is a production of insect resistant plants. Some of the examples of these insect resistant plants are BT cotton, BT corn, rice, tomato, potato, soybean, etc. These are the some of the examples of the insect resistant plants, wherein these genetically modified plants have inbuilt resistance towards these insects. For example, BT cotton. Let us see in detail about BT cotton. BT cotton is the first genetically modified plant that was being marketed in India. It is the first GM crop that is released in Indian market. So let us see in detail how this BT cotton is developed, what are the advantages of this BT cotton, what is the mechanism or what are the techniques of genetic engineering that has developed this BT cotton. First, let us see what does this BT stands for. BT in BT cotton stands for Bacillus thuringiensis. It stands for Bacillus thuringiensis. Bacillus thuringiensis is a soil bacteria. This Bacillus thuringiensis is a soil bacteria which is a rod-shaped gram-positive bacteria. We have different bacteria such as gram-positive and gram-negative, but the Bacillus thuringiensis is going to respond positively to the gram staining, so it is a gram-positive bacteria. Now, how this bacteria is related to the cotton plant? That might be in your mind, right? So BT is known as Bacillus thuringiensis and it is a soil bacteria and it is going to be present in the soil. So in the year 1976 and 1977, Zacharian scientists discovered certain genes in the plasmids of these some of the strains of the Bacillus thuringiensis. He has discovered certain genes, okay, present in the plasmids of these strains of the Bacillus thuringiensis. Now what were these genes known as? These genes were called cry genes. The genes which were studied in the plasmids of the Bacillus thuringiensis is known as cryogenes. And these cryogenes, now every gene, there is a gene present in the body, that is in the genetic material. When this gene codes, then only it is going to form a protein. Similarly, the genes of the Bacillus thuringiensis, okay, known as cryogenes. Why they are known as cryogenes, you will come to know. Okay, these genes have the capacity to code for certain proteins, and these proteins are toxin in nature, they are toxic in nature, and they are called delta endotoxins, or they are known as crystal proteins, or they are known as cryproteins, or they are known as Bt toxins, because they are produced from the Bacillus thuringiensis. And they are known as insecticidal proteins, because the cryogenes are coding for certain proteins which are toxic in nature. They are toxic towards a group of insects. They are toxic towards a group of insects, and therefore they are known as insecticidal proteins. And these are produced during certain phase in the growth cycle of the bacteria, right? The growth cycle of the bacteria has a different phases in its growth cycle, like lag phase, log phase, stationary phase, exponential phase, death phase, right? So during this, at certain point of time in its growth cycle or in its lifetime, it is going to produce these toxins, and that particular phase is known as sporulation. These toxins are released during sporulation of its growth phase, and these proteins act against different group of insects. So each strain of the Bacillus thuringiensis produces certain proteins that act against a specific type of insects. Not all the strains are producing similar type of toxins and they act on all types of insects. No, not like this. Specific proteins are produced by certain strains of the Bacillus thuringiensis, and they are active against only specific target pest or insect. So these proteins act against lepidopterans such as tobacco birdworm, armyworm. It also involves cotton bollworms. Okay, these are the some of the insects that feed on plants. And there are coleopterans, which includes beetles. These toxins are also active against the coleopterans, which involves beetles, and also dipterans, that includes flies and mosquitoes. It includes flies and mosquitoes. So if we see the Bacillus thuringiensis, why the cotton is known as Bt cotton, okay, when the study has been conducted, like Zacharian, when he has tested the genes of the plasmids of the Bacillus thuringiensis, they are coding for certain toxic proteins which are known as delta endotoxin, or they are known as crystal proteins, or they are known as cryproteins. This is a neat point. It is not there in textbook, and these are produced during sporulation, and they are insecticidal in nature. And Bt toxins are specific in their nature, means based on the target insect or pest and type of the crop, specific genes are isolated from the Bt and incorporated in the crop plants. Not all the toxins what I said, they are insecticidal towards all the insects. No, particular group of cryogenes are going to code for certain toxins that are going to act on only specific type of organism. So when the crops are developed with this insect resistance, we are going to study what type of crop it is, what are the insects that are feeding upon these crops, and what are the type of the cryogenes that is going to produce toxins which are going to be effective towards these insects, right? Because almost there are 300 cryogenes are isolated and they are studied, which will code for the crystal proteins. Now all the 300 cryogenes are not active against all type of insects. A particular cryogene will code for certain toxins towards a group of insects, right? So Bt toxins are specific in nature. Then you might be wondering, when the insect resistant plants are produced, and we take the product like cotton will be extracted from these plants and it is supplied. When humans or other mammals like birds and fishes are in contact with these plants, why the bt toxin is not affecting these people or affecting these humans and other mammals? That question would be in your mind, right? Yes, bt toxin will not affect the humans and other mammals because they are produced as protoxins. They are produced as protoxins. Pro means it is a inactive form. Like some of the enzymes in the body, they are produced initially in the inactive form. Then based upon the conditions of the environment, they get activated, like change in the pH. For example, in the stomach, if you see in the intestine, if you see the trypsinogen, if it is produced, how it is produced in the inactive form. Trypsinogen is produced in the inactive form, so it gets activated based on the pH of the surrounding environment. They get activated to trypsin, trypsinogen to trypsin. So similarly, this toxin also, it is released as a protoxin. Therefore, if it is digested by the human, it is not getting the required conditions in the gut region of the humans, and therefore it is excreted out as such without digestion, and therefore it is not harmful for humans, right? Okay, and this bt toxin will not, it is not toxic towards the bacillus thuringiensis also, because it is produced as a protoxin which is going to be inactive in nature, right? Okay, we have almost 300 crygenes, and some of the crygenes are coding for certain toxic proteins which are going to act upon against a group of insects. For example, genes cry1ac and cry2ab, they control the cotton bollworms. Genes cry1ac and cry2ab that controls the cotton bollworms. These cotton bollworms are feeding on the cotton plants. So if the bt cotton is engineered, then the plant is going to contain these genes cry1ac and cry2ab, which will code for the bt toxin, and that is going to act against the cotton bollworms, and it is going to destroy these cotton bollworms when they are feeding upon the cotton plant. Cry1ab controls corn borer. The gene cry1ab gene controls the corn borer, and if these are not there in the textbook, these are once again the neat bits. Cry1ac and cry2ab, it is there in the textbook, which controls the cotton bollworms. Cry1ab controls corn borer. Cry3ab controls Colorado potato beetle. Cry3bb controls corn rootworm. 1ac and 2ab controls cotton bollworms. 1ab controls corn borer. 3ab controls Colorado potato beetle, and 3bb controls corn rootworm. Right, students? So this is about the bt toxins, how the word bt has come. So based on the ideology, since the bt toxins have the power to act against these insects, so using the genetic engineering techniques, these genes, that is the cry genes which are coding for the bt toxin, are introduced into the plants, using the genetic engineering techniques. So the bt cotton engineered using the ideology of bt toxin. Now let us see how the construction of the bt plants is going to take place. How the construction of bt plants is going to take place. Now we know, we have seen certain genes, cry genes 1ac, 2ab, 1ab, 3bb, right? These are the some of the cry genes. Now you know cry1ac and cry2ab, it encodes toxins which control the cotton bollworms. Now in case of bt cotton, to construct the bt cotton, we need the gene cry1ac and cry2ab to be inserted in the plant cell. So the Bt toxin gene first must be isolated from the Bacillus thuringiensis strain, and it must be cloned from the bacteria, and it has to be expressed in the plant to provide insect resistance plants, right? So once the Bt genes are isolated from the plasmids of the Bacillus thuringiensis, what will happen? Now, after you got the gene of interest, if you go back to the biotechnology principles and processes, what will happen once you have isolated the gene of interest? Next is it must be linked to the vector DNA, that is cloning. It has to be cloned. Once it is linked to the vector DNA. So for cloning genes in the plant cells, what are the vectors we are going to see? We are going to use, in the previous chapter you have seen, for cloning genes in plants and animals, we have, in case of plants, we are going to use Agrobacterium tumefaciens, that is a Ti plasmid of Agrobacterium tumefaciens. Or another important method that helps in gene transfer in plant cells, that is the biolistic method or gene gun method. So using the Agrobacterium tumefaciens, if the gene transfer is done, it is known as Agrobacterium mediated gene transfer. It is known as Agrobacterium mediated gene transfer. Or if it is you're directly using inserting the recombinant DNA, that is the CRY genes are inserted in the vector DNA, that is known as recombinant DNA. And similarly, if you are inserting this recombinant DNA using a particle pressure gun, which is known as a gene gun method, which is going to be hit the nucleus of the plant cell with very high velocity and insert the microparticles, right? The microparticles are made up of gold and tungsten, are coated with your recombinant DNA, which is your vector, and it is a gene of interest, right? So based on these two, so if you are using Agrobacterium tumefaciens, okay, whose Ti plasmid will contain the gene of interest, CRY1AC and CRY2AB genes. The Ti plasmid contains a particular DNA known as transfer DNA, which is known as tDNA, which is going to convert the normal cells into the cancerous cells, right? So the disarmed pathogen, it is used to transfer the DNA. So here CRY genes are inserted in the Ti plasmid, and this Ti plasmid, it can be inserted into the Agrobacterium, and Agrobacterium is allowed to infect the plant cells. If you see here, this is the Ti plasmid of the Agrobacterium tumefaciens. Here Ti plasmid is present, and here the chromosomal DNA is present. Okay, this circular double-stranded DNA is the Ti plasmid, and this Ti plasmid contains a tDNA here. You can see the shaded region tDNA, and this tDNA, this is the tDNA, right? Now this tDNA is getting attached with the gene of interest. This is a DNA containing gene for the desired trait, which is the CRY1AC and CRY2AB genes are present, right? Now these both are ligated. First, the restriction enzymes are used to cleave the tDNA, and you are going to using the DNA ligase, we are going to insert the CRY genes here. We are going to insert the CRY genes in the Ti plasmid in the region of tDNA. Here we have, this is a recombinant DNA containing the Ti plasmid with the tDNA along with your gene of interest. The red color region is your gene of interest containing CRY1AC and CRY2AB genes. Now you can directly using the gene gun method, this Ti plasmid, the recombinant Ti plasmid, can be inserted into the plant protoplast. Okay, that is a plant protoplast which contains the cells. It is a part of the plant, and this is this tDNA containing now new gene with the plant chromosome. Together it is going to contain, and using the tissue culture, these plant cells are regenerated using the tissue culture grown in the artificial nutrient medium, and the plant is developed. This plant contains the desired genes, that is the Cry1AC and Cry2AB, which is going to code for the Bt toxins, which are insecticidal in nature and which is going to provide resistance towards the insects, especially in case of cotton. It is resistance to the cotton bollworms, right? This is how genetically modified Bt cotton plants are produced. Let us see now mode of action of Bt toxin. How the Bt toxin is going to act in the Bt plant. Now, though you have constructed, you are introducing the genes in the cotton plant. But how these genes are expressing? Whenever the gene is expressed, then only the protein is going to be produced, right? So, the Bt cotton plant now contains the Cry1AC and Cry2AB genes. Okay, when it is going to be placed along with the promoter gene, which is going to regulate the expression of these genes to form the Bt toxins. Okay, so in the provided conditions, the Bt genes, that is the Cry genes, are going to code for the Bt toxins, and these Bt toxins are produced in the plant as inactive protoxins. They are produced as inactive protoxins, and whenever the insects, in case of Bt cotton plant, they are the cotton bollworms. When they are feeding on these Bt cotton plants, these inactive protoxins are ingested by these cotton bollworms. When they feed on these cotton plants, they eat up the leaves of the cotton plant, which contains these inactive protoxins. Now, whenever the insects are ingested with these inactive protoxins, okay, when they pass through the gut region, what will happen? Due to the alkaline pH of the gut region of the insect, is this inactive protoxin is getting converted to the active toxin. It is getting converted to the active toxin. Now, this active toxin, okay, is going to act upon the cells of the insect. Getting the alkaline pH, whatever of the gut region is present, is going to solubilize the crystal protein and activates the toxin, and this toxin is going to bind to the surface of the midgut epithelial cells through specific receptors, and it is going to produce certain pores which is going to cause the swelling of the cell. It is going to cause the pores are present, okay, which is going to cause the swelling of the cell, and this, after the cell is completely swollen, it is going to lead to the cell lysis. When the cell lysis is taking place, the contents of the cell gets leaked out and the toxic substances are spread towards the whole part of the insect, and that is leading to the death of the insect. First, these are produced as an inactive protoxin, and then when it is ingested by the insects, due to the alkaline pH of the gut region of the insect, the inactive protoxin gets converted to the active toxin, and the active toxin binds to the midgut region of the epithelial cells through specific receptors, which creates spores that causes the cell swelling and leading to the lysis of the cell, which is going to the infection is spread towards all the parts of the insect and leading to the insect death. Therefore, this is how Bt cotton plants are going to produce the insecticidal properties towards the insect, and therefore there is reduced reliance of chemical pesticides because the plant itself is providing resistance towards these insects. Clear? So this is one of the major application of biotechnology in agriculture, that is production of the insect resistant plant. Now let us see production of pest resistant plants. We have seen now production of insect resistant plant. So in application of biotechnology in agriculture, we will see production of pest resistant plants. What are the pest resistant plants? So there are the certain pests are which are feeding on the plants and they are destroying the plants, that is leading to the crop loss. It is leading to the crop loss. For example, parasite nematodes. Parasites, you must be knowing what are parasites. Parasites which are the living organisms that is going to be feed on the other plants or other living organisms for its growth and development. They live on other organisms for their growth and development. They are known as parasite. So, some of the pests that are feeding on the plants are the nematodes, which are known as roundworms belonging to phylum Nematoda. So, therefore, they are known as parasite nematodes. Now, these parasite nematodes are going to be contributing to the major crop loss. Okay, and one of the species of these parasite nematodes are the Meloides incognitia. These are going to contribute to the major crop loss, that is in the tobacco crops. It is going to create damage in the tobacco crops. Some of the nematodes are parasite in nature and they feed on plant roots. They feed on plant roots and cause the roots to grow large and form tumors or knots. This is how they are going to damage the crop. Okay, these parasite nematodes, especially the Meloides incognitia, that is a roundworms, they are going to feed on the roots of the tobacco plants and they are going to cause root knot disease. That is, it is going to swell the roots and cause the tumors and knots. Therefore, the nutrients from the root cannot be absorbed or transported to the shoots and therefore that is leading to the damage of the crops. Right? This is how the economy loss can take place of the country. Therefore, again the technique of biotechnology, that is one of the important technique is being implemented, that is going to prevent this type of parasite nematodes destroying the plants, and that technique is known as RNA interference. RNA interference. What is this RNA interference? This is one of the technique of the genetic engineering once again, that is silencing of a specific mRNA due to complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA. Students, don't get confused. I have already explained to you people the basics of the genetic material. Genetic material DNA is known as a central dogma. Why? Because it is going to undergo 3 different types of processes which you people will learn in detail in the molecular basis chapter. In the molecular basis of inheritance, there you are going to learn the about this genetic material DNA in depth. Okay, but for time being you should know what are the different processes wherein the DNA is involved, and in order why it is known as genetic material and why it is so important, because it helps in the cell division, it helps in the cell growth and everything, right? Because it is going to produce certain proteins required for the cell. One is it undergoes DNA replication. Okay, that is DNA replication, and then once the DNA is replicated, it is transcribed, means the DNA is getting converted to mRNA. There is a messenger RNA, right, which is going to send the message to initiate the protein synthesis, right? That is known as transcription. Later this mRNA will be converted to the protein, that is known as translation, means mRNA is translated into protein, that is known as translation. Clear, students? So RNA interference is silencing of this specific mRNA, the mRNA which is going to produce the protein, because proteins will have certain functions that helps, either they act as hormones, they act as enzymes, and it is going to produce certain products which are required, right? So silencing of a specific mRNA due to complementary double-stranded RNA molecule. Getting? So to the mRNA, whatever is present, it's complementary strand, what is known as double-stranded RNA molecule. it is going to bind and it is going to prevent translation of the mRNA. The mRNA is going to be silenced by the binding of the double-stranded RNA. Getting? If the double-stranded RNA binds to this, or the double-stranded RNA contains a particular complementary strand to this mRNA, which is going to prevent the translation of this mRNA. If the mRNA is not translated to protein, then the proteins are not produced which are required by the parasite, and therefore the plant cannot utilize these proteins, and that will lead to the death of the parasite. Clear, students? So this RNA interference, it is also known as gene silencing, or it is known as quelling, or it is known as co-suppression. Very much important neat bit. This RNA interference, it is also known as gene silencing, or it is known as quelling, or it is known as co-suppression. You can also observe this RNA interference in eukaryotes as a part of the cellular defense mechanism. It is known as cellular defense mechanism. There you can observe RNA interference. Means if any organism is invaded, okay, if any organism is entered into the body, then the body is going to defend against that organism by RNA interference. So whatever the mRNA that is produced by these organisms will not be able to translate their proteins required for their development by the RNA interference. Clear? So the RNA interference is taking place in eukaryotes as a part of its cellular defense mechanism, and also it is used in gene regulation process. Gene regulation, like now how the gene regulation, gene expression is going to take place. A particular gene will code for a particular protein. How? Once again the gene will get replicated. After that transcription will take place, and then translation will take place. So the mRNA whatever is formed, when it is translated, it is going to form the product. Okay, how it is regulated? The product must be formed only when there is requirement. Excess production of the protein or product, what will happen? Will lead to the abnormality. So to prevent that, the gene expression must be regulated. So how this once again the prevention of translation must take place? It is once again by RNA interference. So RNA interference is observed in eukaryotes in terms of cellular defense mechanism, or as well as a part of the gene regulation process. Okay, so in order for the silencing the specific mRNA, we need a complementary double-stranded RNA is required. We need a complementary sequence of mRNA or the double-stranded RNA is required for suppressing the specific mRNA, which is known as antisense RNA. Because mRNA will code for a protein, so it is known as sense RNA. And the complementary strand to this mRNA is known as antisense RNA. Whenever in the protein translation process, okay, if there is an antisense RNA is produced, then their protein will not be formed, so it is known as gene regulation process. Means the mRNA, that is a sense RNA, and if the antisense RNA is produced, since they are complementary to each other, they bind to one another. The complementary base pairing will takes place, and it forms double-stranded RNA, and double-stranded RNA will initiate the RNA interference, and the protein will not be formed. Right? So what are the sources of this complementary RNA? From where you can get this complementary RNA? You can produce it synthetically or artificially by transcription from a single-stranded DNA. From a single-stranded DNA, we can produce the complementary RNA by transcription process, or by infection by viruses having RNA genome, if a virus having a RNA genome and it is going to infect, then once again you can produce a complementary RNA can be produced, and genes, mobile genetic elements which are known as transposons, or they are known as a jumping genes, they also, which are produced via RNA intermediate, will act as complementary RNA. So for the main basic thing for RNA interference is double stranded RNA. That is double stranded RNA. If the double stranded RNA is present, that will initiate the RNA interference process, and it is going to silence the specific mRNA can be silenced. Now let us see how this RNA interference is going to takes place. How the RNA interference is going to takes place. What we required first is a double stranded RNA. So therefore, if you want to produce a parasite nematode resistant plant, the plant should have the genes that is going to produce the complementary RNA, because the parasite nematode will produce the mRNA. So we are going to silence the mRNA of the parasite nematode. So the complementary RNA for that mRNA must be produced by the plant. Yes or no? Therefore, the plant must be introduced with the specific genes that can generate sense RNA and antisense RNA. Because in order to initiate the double stranded RNA, we require sense RNA and antisense RNA. The sense RNA and antisense RNA together will form the double stranded RNA, which is going to initiate the RNA interference pathway. Okay, so we are going to introduce certain specific genes in the plant, that is the tobacco plants, which are going to be containing the genes that will code for the sense RNA and antisense RNA. Since these two RNAs are complementary to one another, it will form the double stranded RNA. It is going to form the double stranded RNA. Right. Now, this double stranded RNA first will be acted by an enzyme known as dicer. There is an enzyme known as dicer. Okay, it is going to be acted by the enzyme dicer, and that will be cleaved into short segments of the double stranded RNA. The long double stranded RNA will be acted by the enzyme dicer, which is a RNase 3 enzyme. It is a RNase 3 enzyme, and which is going to cleave the long double stranded RNA into small segments of double stranded RNA known as siRNA. So these are the small segments of the double stranded RNA known as siRNA, that is short interfering RNAs. It is known as short interfering RNAs or small interfering RNA known as siRNA. Now this siRNA will complex with a protein group to form RISC complex. Okay, you can see here the circular ones. Okay, this is known as the RISC complex. The double stranded short interfering RNA will form or combine with the protein complex known as RISC complex. This RISC complex is known as RNA induced silencing complex. It is known as RNA induced silencing complex. Right? Once it is forming the complex with the RISC protein group, the siRNA will unwind and form ssRNA. You can see here single strands of the RNA are present, that is known as the ssRNA, that is single stranded RNA are formed when it is attached to the RISC complex. Unwinding of siRNA to ssRNA is going to take place, and then now the single stranded RNAs will contain the sense RNA as well as the antisense RNA. The blue colored one single stranded RNA is known as sense RNA, and the green colored ones are known as antisense RNA. This is the antisense RNA, which is complementary to the mRNA, that is messenger RNA. Getting after the unwinding, okay, the RISC complex now contains only the antisense RNA. The sense RNA gets degraded and only the antisense RNA is produced. Only the antisense RNA is produced. So this is the antisense RNA. Now, the binding of the target mRNA to complementary or antisense RNA is going to take place. After the RISC complex binds to the antisense RNA, this antisense RNA activates the RISC complex, and now it is going to find its complementary mRNA or the messenger RNA. Means the antisense RNA is complementary to the mRNA, right? So this is the antisense RNA. Now it is in search. It gets activated here, and now it is in search with the messenger RNA. That is the blue colored one is the mRNA. So after it gets activated, it binds to the mRNA. Here you can see it is bind to the mRNA, and it contains protein known as Argonaute-2 protein. It contains Argonaute protein, which is going to cleave the RISC complex contains the Argonaute protein that is going to cleave the mRNA into the small segments. It is going to degrade the mRNA into the small segments. When mRNA is not there, it is not going to translate into protein, and therefore it cannot translate into protein, and therefore the parasite nematode cannot get the required proteins, and it leads to the destruction of the parasite because there is no expression of gene or the translation is inhibited and no proteins are synthesized. This results in the death of the parasite nematode in the transgenic host expressing specific interfering RNA. Means the parasite nematode cannot survive in the transgenic host which is expressing specific interfering RNA. I hope it is clear, students. Initially double-stranded RNA will be present, so it will be acted by the dicer enzyme, and then long double-stranded RNA converted into short interfering RNAs, and this short interfering RNAs get attached with the RISC complex which are circular in nature, and the protein in the RISC complex is going to unwind the short interfering double-stranded RNA into the single-stranded RNA which are blue and green color in nature that contains the sense RNA as well as the antisense RNA. Green colored ones are antisense RNA and blue colored ones are sense RNA. Right? Sense RNA gets degraded and antisense RNA along with the RISC complex. RISC complex is RNA induced silencing complex which contains Argonaute proteins. Now the RISC complex antisense RNA activates the RISC complex, so they go and find the complementary mRNA. Okay, once it finds the complementary mRNA, the antisense RNA binds to this complementary mRNA, and Argonaute protein present in the RISC complex is going to degrade the mRNA into pieces, into very minute segments. No more it is going to be functional, and therefore when mRNA of the parasite is destroyed, there is no expression of gene or the translation is inhibited and no proteins are synthesized. This results in the death of the parasite nematode in the transgenic host. Clear?